Welcome to our digital representation of the Mycoplasmas, a group project by Louis, Maria and Alexandra. This is a video presentation to help you navigate our website. At the bottom of the page you'll find a button that says Taxonomy and Physiology. This represents the contents page as well as the introduction. Under the taxonomic description, you'll find the phylogenetic tree depicting every Mycoplasma species that humans have classified. Mycoplasmas are known for their miniature genomes, consisting of only 500 to 1,000 genes, which is the smallest we know of. This page focuses on the general techniques that the organisms employ and their core features. Firstly, you need to understand that mycoplasmas are parasitic, meaning they adhere to the host cell and use its resources for its own survival. This is always disadvantageous to the host, so we do not consider it symbiosis. Mycoplasmas evolved to reduce their genome size as much as possible, and they are especially hard to research on media in a lab. Here is a diagram of the capabilities of mycoplasmal adhesion, which shows how we can alter factors in the host cell to ensure the survival of parasitic mycoplasma. Here we have engineered contents page into a section about physiology. You will be able to select features that interest or confuse you to relocate you to an explanation. The Mycoplasma are the class which represents a lot of the Mycoplasma's distinguishing traits. It is, however, an umbrella term for gram-positive bacteria without a cell wall. This can often confuse gram stain results, as there is a lack of peptidoglycan to retain the safranin and crystal violet. Instead, the organism has a three-layered membrane held together through hydrophobic forces as well as structural integrity created by lipoproteins and sterols respectively. Sterols are unique in this case, as it is the only bacteria that requires them, and if you change the concentration of fatty acids in the media, then the growth of the membrane changes significantly. The virulence of mycoplasmas, or the way in which the organism imposes itself onto its environment, has to be specialized, as the physiology of the bacteria is quite unique. This section depicts three main areas of importance. Firstly, we explain the function and structure of catalase to remove free radicals, and how mycoplasmas work to inhibit this in their host cell to reduce the cell's functionality. Secondly, how adhesins work to bind and stick to receptor proteins on a host cell's membrane. We also go into detail on how this suppresses the immune system and the detection of a parasite. Lastly, this page introduces the idea of how mycoplasmas can help in the inadvertent development of AIDS through inflammation of the genitals, allowing HIV into the subepithelial cells and progressing the disease rapidly.
Hi guys, so my name is Maria and I will be speaking to you today about specific species within the Mycoplasma genus. So I've started with some general information. So mycoplasmas have very limited biosynthetic capabilities and they use host cells to multiply and survive. It's almost parasitic-like, kind of like viruses. They belong to the class Mollicutes and family Mycoplasmataceae. They have single triple layered cell membranes which include sterols. These are required for growth and they help the structure of the cell membrane, as mycoplasmas do not have cell walls. Mycoplasmas adhere to cells in the respiratory and urogenital epithelium as an initial and essential step in tissue colonization and disease pathogenesis. So I've got an example of Mycoplasma pneumoniae, which has a tapered tail and specialized proteins that help it adhere to cells. So yeah. So I have started by explaining Mycoplasma pneumoniae, which is a rod-shaped strict anaerobe found in the respiratory tract. It's got a length of 150 to 200 nanometers. It can inhibit catalase in human cells, and it also has the tapered tail, as I said before. Then we will go on to ecology of Mycoplasma pneumoniae, and you can see a picture which has pointers to the tapered tail, and you can see the shapes of the Mycoplasma pneumoniae. The shapes change as this is a pleomorphic usually. It can change its shape quite easily since it doesn't have a cell membrane. Um, they are not found free in nature as they require a host cell to survive. They can be grown in an artificial growth medium, but must have sterols for their triple layered cell membrane to be formed. Yeah, as I said, they have no rigid cell wall, which means they can change shape quite easily. And the surface proteins allow adhesion and entry into host cells, which you can see by the pointers on the picture. Okay, so next I will move on to Mycoplasma hominis, which is a pleomorphic bacteria with lengths of around 0.2 to 0.3 micrometers. It's an aerobe, usually recovered from the lower genitourinary tract, also known to thrive in the respiratory tract of humans. It can be transmitted sexually, and the body's submucosa usually prevents problems caused by Mycoplasma hominis. It's the smallest bacteria capable of self-replication, and it uses UGA to code for tryptophan instead of a stop codon, which is what it would usually code for. Then we go on to ecology, and as you can see, there's a picture of Mycoplasma hominis, and people usually say it looks like a fried egg, and it's brown apparently under a microscope. So it's known to live in human and primate urogenital tracts. It creates a metronidazole resistance when symbiotically related to Trichomonas vaginalis. So it can survive for up to 1 hour in liquid specimens if protected from evaporation, and it can be found in toilet bowls. So next we'll be talking about Mycoplasma genitalium. This is a pear/flask shaped bacterium with a terminal tip organelle. This terminal tip assists attachment to various surfaces and provides gliding motility. So yes, this bacterium is motile. It has a genome of 580 kilobases and a comparatively higher GC content than other mycoplasmas. It is one of the smallest prokaryotes capable of replication, and it's an established cause of non-gonococcal urethritis in men. So if you would like to stain this and so it's visible, DNA fluorochromes, for example, acridine orange stain, may be applied to body fluids after centrifugation. There is no reliable medium to culture this species, but it can be made in cell cultures, although this is time consuming yet effective. It can also be detected by PCR. Next, we move on to ecology. You can see the picture of the Mycoplasma genitalium with the terminal tip clear. So the main adhesin on the terminal tip has been identified as the MGPA protein. This encodes for P10 and P40, which are site adhesins. So the loss of these proteins would result in no motility and a lack of adherence to cells. This can show antigenic variation of surface components to evade the host immune systems, and can also mimic hosts' antigens to survive. So it's almost like tricking our immune systems, and like putting on a wolf putting on sheep's coat. Yeah, so it basically hides itself in our immune system, and our immune system cannot detect it as foreign. Lastly, I will be talking about Ureaplasma urolithicum. So this is not necessarily part of the Mycoplasma genus, although it is the closest genus to Mycoplasmas. It is also a class of, well, a group of different mycoplasmas that are found in the genital tract. So, yeah, I thought I would still include it as there are quite a few similarities, mainly similarities to mycoplasmas, but there are a few differences which I will highlight. So the shapes range from filamentous to spherical, so it's pleomorphic. It has a diameter of up to 0.8 micrometers. It has no cell wall but a triple layered membrane including sterol. It has urease, which converts urea to ATP plus ammonia. The ammonia increases pH and limits growth. If you would like to see this, well, grow this bacterium, incubation in 100% carbon dioxide will result in larger colonies than nitrogen, oxygen, and air. Also, if you add putrescine and urea to the growth medium, you also get improved growth. So, ecology, it grows around pH 5 to 9. It's usually found in the vagina as it's a common cell that often does little harm. It's usually isolated from the genitourinary tract in humans, and it's also isolated from amniotic fluid. It only causes infections when it travels up to the upper reproductive tract, and that results in adverse effects on pregnancy. So yeah, you can see a picture of the ureaplasma next to the words. And yeah, thank you.
My section focused on the pathogenesis of mycoplasmas. So most mycoplasmas invading humans and animals are surface parasites, and they're often found on the mucosal surface of the respiratory and urogenital tracts. There's 7 species of mycoplasmas that are found to be pathogenic in humans, and well, they're listed here. And most mycoplasmas depend on adhesion to infect host cells. So once they adhere to the host surface, it can be really difficult to remove them through mucus secretions or urine. And adhesion also allows toxic metabolites to be excreted by mycoplasma, and it generates a lot of tissue damage. And because of the absence of a cell wall, mycoplasma often fuse with the membrane of the host cell, and this allows hydrolytic enzymes to enter the host cell and cause further damage. So I've chosen to specifically focus on the pathogenesis of M. pneumoniae because it's one of the most well-examined species of mycoplasmas. This section is just basically a very brief overview of what I'd be covering in the next sections. So if you want to find out more detail on direct damage mechanisms, immune damage, inflammatory damage, and antigen immune damage, you can click on the buttons. Okay, this section covers direct damage mechanisms. So if you scroll down the page, you'll see the subheadings: adhesion damage, membrane fusion damage, and toxic damage. I won't go into too much detail, but first of all, adhesion involves a really important protein called the P1 protein, because without the presence of this protein, M. pneumoniae can't adhere to the host cell. And secondly, mycoplasma lack a rigid cell wall, so often they fuse with the host cell membrane, and this can cause changes in the nucleolus and receptor sites. In terms of toxic damage, Mycoplasma pneumoniae can release hydrogen peroxide, which causes further damage to respiratory cells, and it also activates lymphocytes, plasma cells. It also produces an exotoxin called the CODS toxin that stands for the community-acquired respiratory distress syndrome, and it basically aids in the colonization of Mycoplasma pneumoniae, and it leads to inflammation and airway dysfunction.